Muy buenas tardes a todos. Me da muchísimo gusto estar en otro de nuestros seminarios del Instituto de Ecología y me da mucho más gusto y gran emoción poder presentarles al doctor Aaron Shafer. Él es profesor en la Universidad de Trent, en Canadá. Su trabajo involucra el uso de herramientas moleculares y genómicas para estudiar una enorme diversidad de aspectos evolutivos y ecológicos. Estudia organismos básicamente en su medio natural para entender procesos, historias poblacionales de estas diferentes especies animales. Y entre las preguntas de investigación que él aborda y en su laboratorio está evolución fenotípica, reconstrucción de eventos demográficos e identificación de mecanismos moleculares que regulan la variación fenotípica particularmente. Pero sus intereses son muy amplios y también incluyen la aplicación de estos métodos genómicos en múltiples preguntas de conservación y en temas forenses. En realidad, de hecho, su laboratorio está afiliado con uno de los programas, es el de Forensic Science. También está involucrado con el Environmental Life Sciences, otro departamento, y el departamento de Applied Modeling and Quantitative Methods. El doctor Aaron Shafer también trabaja mucho en cuestiones de modelado y aspectos matemáticos. Pues aquí está el título de su tesis y de su presentación. Y sin más, le doy la palabra. Aaron, very many thanks to be here and to have accepted your virtual talk, and I promise we will bring you Mexico sometime.
Thank you, Ella, and thank you for the invite, and I will take you up on that. I had this in my calendar last year, and I was looking forward to coming to Mexico City, but hopefully next year things seem to be improving. This is a new talk for me. It's brand new, and I've titled it Wildlife Genomics, Moving Beyond SNPs and Outliers. And what I want to sort of talk about today, I want to give you a bit of a background why I think we might need to move beyond SNPs, why we like looking at SNPs and outliers and adaptive genes, but why also we might need to be thinking a little bit beyond that. And I'll tell you that story, and then I'll bring in some of my research where we're trying to move beyond sort of traditional SNP analyses. And so to begin, when we talk about SNPs, we have to remember what is in a genome. And you know, we're all in this online environment, we're diploid organisms. So we get a chromosome from our mom and our chromosome from our dad. And when we look at variation or SNPs, we can see variation just in my genome. So if my dad gave me a T and my mom gave me an A, just looking at my genome would show variation. But I can also compare my genome to your genome, and we can see over here, there's a T and a G. And so that's really the basis of identifying SNPs. We then take this variation and we start to ask questions about maybe, you know, our ancestry and, you know, what were the colonization routes out of Africa using genomic data, or looking at phenotypes and trying to find mutations or SNPs controlling for variation in phenotype. And this, I think, it would be fair to say, has been the focus of wildlife genetics, population genomic research for the past 20 years, and at least the past decade with respect to high throughput sequencing. We've been really focused on identifying these variants and asking questions with them. But there's a lot more in a genome. And that's what I'm going to talk to you a little bit about today. Now we're seeing a lot of value in looking for, say, for example, structural variants. So where we have massive insertions or deletions. And we have a great example of a crow hybrid zone in Europe. This was my postdoc lab, where it seems that what sort of maintains speciation between the hooded crow and the carrion crow is really a single insertion of a jumping gene. The rest of their genomes are virtually homogeneous, but then you have this divergence underlying speciation. So we're seeing structural variants, and more now we're able to quantify things like methylation and epigenetics. And so we often think of DNA methylation, maybe 1% of our genome is methylated. So if a human genome is 1 billion base pairs, 1% is a lot of information that we can look at. but we can also look for things like histone modification. And these are just two examples of sort of additional things that we're now starting to look at when it comes to genomics beyond SNPs. And then there's much more than these two, but I just wanted to highlight that we're learning more and more. But I think it's worth asking the question, why have we focused on SNPs for so long, and why is this the focus of SNPs? Why is the focus of wildlife research been on SNPs? And I think probably the easiest answer is that it works well in humans. We do SNP research in humans all the time. And maybe by show in the chat, if you don't mind sharing, has anyone had a genetic test, either personal or through sort of a doctor? I've had both, so I've done 23andMe, and I've also had, my family has a hereditary condition, and so if you feel like sharing, I would suspect some of you have. And some of you have because we know we can reconstruct your ancestry very well, and there are some really good predictive assays with respect to various diseases. So in part, the focus on SNPs has been because we do it in humans all the time. It makes sense and it's very useful. In addition, it's pretty easy to do. And so pre-Illumina, we could identify SNPs using Sanger sequencing or cloning. And you can see here, you can pick up the variant pretty easily with these overlapping peaks in the electropherogram. Pretty easy assays to design. We can also do qPCR. My lab does a little bit of this when it comes to validation. But again, so we have homozygotes in blue and red and heterozygotes in green, and SNP chips. And so it's pretty easy to quantify SNPs. So that's another reason that we've had this sort of fascination with SNPs for the past 20 years. And there are lots of examples of those SNPs being informative. And so if you were to design a study and you had phenotypic variation in coat color, pretty much any vertebrate, you'd probably want to look at the MC1R gene. And you can do that. And we know we've well characterized this, where single mutations that cause amino acid shifts can have dramatic consequences when it comes to the phenotype. And this is pretty true across vertebrates. And we even know in some sheep that this affects behavior and dominance variation in this gene. There are lots of examples in the human world. There's tons. And I just pulled this one up because I thought it was fascinating. It shows you how efficient we can do this in humans. This study's from March 2020 with respect to genomic associations to COVID-19 critical illness. And so, in North America, right, March 2020, in Canada, we hadn't gone into lockdown yet. UK, we were watching it unfold largely in Europe, but the human data sets are so efficient that they can immediately screen genomes and identify risk alleles when it comes to COVID. So there are lots of examples of our focus on SNPs. And I think from the wildlife context, there's been this idea that SNPs will be the panacea when it comes to conservation genetics and conservation biology. That through sequencing genomes, identifying variants, all of these unsolved conservation problems highlighted here in a lot of review studies argued that genomes, and in particular SNPs, will be the way forward. This is how we're going to conserve our species that are in trouble. And I like this slide, and I always highlight this, that all of these review articles talking about genomics, talking about SNPs and conservation, they predate 2010. And 2010 for me is a very important year because that really marks the entry of high throughput sequencing into wildlife research. Prior to 2010, it was largely restricted to humans and model organisms. And so what that means with respect to these articles is none of these articles, none of these sort of arguments for SNPs are based on empirical data when it comes to conservation. And it generally revolves around, if we have more SNPs, we'll be able to have better estimates of demography, hybridization, conservation units, viability, so on and so forth. And generally that's true, but the idea and the argument has always been more markers, more SNPs, allow us to answer these questions and identify adaptive loci. And I think that is why we see a huge interest in SNPs. As well, it's a story about technology. And so we know, at least in the conservation literature, that when new techniques emerge, maybe it's GPS radio collars, genomes, we see a huge uptake in the conservation community. But then over time, we see that there's a knowledge gap that emerges. And I think this is partly this partly explains why we've seen such interest in SNPs in wildlife genetics, because now we can, and we think and we hope that it's useful for conservation. But why we might not want to focus on SNPs in wildlife is an important question. And this is something I've been thinking about for a while. And there are some valid reasons why we might question the use of genome sequencing and SNPs when it comes to conservation. And one of them is sample size. Wildlife is not humans. And so we don't have the ability to rapidly produce a COVID study that identifies genetic variants. And for example, Marty Cardos showed rather elegantly, if you were interested in adaptation and identifying the genetic basis of a trait, what he showed us was that with 81 genomes, and our x-axis here is effect size, what this means is 0.1 is 10% of that trait can be explained by a mutation. 0.2 would be 20%. It's a huge effect locus. And what Marty showed us was with even 81 genomes, our ability to identify, our power to identify these loci was minimal. And in fact, you would need something on the order of 243 whole genomes to reliably pick up these low to moderate effect loci. If you have a SNP chip, you need SNP chips that are 500,000 SNPs to reliably pick up these loci of small to large effect. And so this should be concerning to you if you're running a study, because most of us can't afford 243 genomes. And in addition, most genes that we know that control traits are in this vicinity, many genes of small effect. Very few genes do we know of that we've detected have a large effect. So most traits, I think, it'd be fair to describe, are controlled by many genes of small effect. In other words, we can't pick them up with our current sample sizes. Marty showed us this in 2016, and my former collaborator in Sweden, Reto Burri, was a co-author on this paper, and he showed the exact same thing with the MHC gene. A lot of us like looking at the MHC gene. Variation is thought to be good, but it's the same story here. We don't have the correct sample sizes. Small to moderate effect loci are largely associated with insufficient power. We can't pick them up. We need sample sizes greater than 200. These are difficult sample sizes to acquire in wildlife, and in a conservation setting, almost impossible. In addition, our genomes, our wildlife genomes, aren't nearly as well annotated as the human genome. And this is important because when you look at humans, we know that 90% of those SNPs that are associated with traits and have been validated are non-coding. In addition, most of these SNPs are pleiotropic, meaning they affect multiple traits. And so when you're thinking about this in a conservation setting, how do you control or conserve a locus that doesn't code for anything as well, or we don't know that it technically doesn't code for a protein, and it affects multiple traits? This is a difficult problem. And on top of that, what I've shown is we don't know how to filter our data. Or if we do it in a variety of ways. And so the way we filter our SNP data, and it's not microsatellites. Microsatellites, we know, Hardy-Weinberg equilibrium, LD, we have these defined filter parameters. With SNPs, we have none of that. And what we showed in 2017 is depending on how you filter your data, perhaps Ella and I have the same data set, but my lab uses one program and Ella uses another program. it can have dramatic consequences on metrics, for example, as FIS. You go from highly outbred to highly inbred would be your interpretation. Population size, we go from a bottleneck to 0 to an expansion of 20-fold just by how we filter our data. And so this is a problem we still haven't yet addressed when it comes to SNPs in wildlife, is how do we properly assess our data? And then the question becomes, what are we actually measuring with our wildlife SNP data sets? And this was a debate that many of you might be familiar with in the literature called breaking RAD, but this idea of looking for outliers with RAD-seq data. And what David Lowry and others showed us is that unless you have massive linkage blocks, meaning you have a highly inbred population, when you do a genome-wide study, in this case RAD-seq, you never pick up the entire genome. So in this case, with sort of average size linkage blocks of 250 or 1,000 base pairs, meaning recombination is shaking things up, unless you have a really small genome, which wouldn't be the case for mammals, you never sample more than 20% of the genome. And so what are you actually measuring? When you get an outlier from a study like this, what is that exactly? How do you trust that that's not a false positive? And this is a very real problem. And collectively, I think these are interesting and fun academic questions. I like the challenge. I like pushing the envelope when it comes to what we can and cannot do with SNPs. And I've written a couple papers, and this is why I typically get invited to seminars, talking about the challenge with respect to genomics and SNPs when it comes to conservation. But I think there really is a challenge and there is a problem, and we need to think about the qualitatively novel aspects of genomes when it comes to conservation and wildlife. If we just run structure, why do we need whole genomes? You know, studies have shown us we can just use microsatellites. And so it's not the numerical value of genomes. More SNPs isn't always the answer to our question. It's the qualitatively novel aspects of genomes. And so I've been sort of challenging the usage of genomics in conservation, or at least questioning it. But again, I'll tell you, I do think that these are fun academic problems. But when it comes to real world conservation and investing money in genomes for conservation, I do think we might need to question that. And it's a bit of a cliche, but the money is probably better spent buying land than it is sequencing genomes when it comes to conservation. So that's the world we're in right now with respect to SNPs. And so I've been trying to make an effort to move a little bit beyond SNPs and our detection of outliers, which I think is quite a common approach. And so what I wanted to do today was tell you about two areas of research where we're trying to think a little bit differently. And one is using SNPs, but here we're using SNPs to generate pedigree-free animal models to estimate heritability. As well, we're also doing pooled approaches. And these are both sort of cost-saving measures that I think can be applied to a lot of studies in the wild and are very informative. And then I want to show you some interesting work about DNA methylation. And then if time permits, I will tell you a little bit about cheap genomes and emerging metrics. And I don't know the funding situation that well in Mexico, but in Canada, we don't get massive grants. And so it's a bit of an obsession of mine, is trying to come up with cheap and efficient ways to generate genome-wide data. And there are some ways to do that. I just wanted to share one of them because it was pretty new to me and I wasn't aware of it not too long ago. And maybe some emerging metrics that I think are pretty powerful, perhaps with conservation if we have time. So now part one, this is working with SNP data. And I want to tell you a little bit about some research we've been doing on deer. Deer belong to the genus Otocoileus. You have them in Mexico, you have both species. Where I am in Ontario, we only have the white-tailed deer. And so we're interested in understanding speciation history of white-tailed deer, but also we're interested in understanding the genetic basis of traits, and specifically antlers and body size. And so my group has been doing work trying to understand the heritability and identify the genes underlying antler variation as well as body size. In addition, mule deer in particular have some pretty extensive migratory behaviors. And so, you know, it's not uncommon for various populations to have migrations on the order of hundreds to 200 kilometers. Nothing like African mammal migrations or caribou for that matter, but pretty extensive migrations. And I'm interested in understanding the drivers of this with respect to the genetics. And so our two main questions with respect to deer are these traits heritable? Things like antler, body size, migration. And you can look to the literature and you have a pretty good idea. Yes, they are heritable, but we want to quantify that and estimate how much of that trait is heritable. And then once we know things are heritable, we want to characterize the genetic architecture underlying these traits. These are both important questions. Once you show something is heritable, you can then predict the response to selection, for example, say from harvest or climate change. And once you know the genetic architecture of a trait, you can develop assays, you know, to measure selection, and even from a practical standpoint, perhaps be informative to breeding and wildlife monitoring with cervids. So this first question, are these traits heritable? And so what we've been doing to ask this question is taking a slightly different approach. If you're familiar with heritability in wildlife, it generally has relied on pedigrees. And these are next to impossible to generate in wildlife mammals. There's a few in wildlife mammal populations. There's a few good examples of cervids, but it's quite intensive because you need to find the mother, you need to find the father, and develop multiple layers. But what we've seen, and this started in the human world, was the use of what are known as genomic relatedness matrices. And really this is a relatedness matrix. And so I take everybody that's attending this talk and we build a matrix. 0 means we're completely unrelated. 1 means you and I are identical twins. But the nice thing is we can generate these genomic relatedness matrices from genomic data, whether it be RAD-seq, whole genome. You can even do it with microsatellites if you wanted. And the important thing here is there is a relationship between this GRM and the sampling variance of our heritability estimates. And we can use that information to actually estimate heritability of the trait using the genomic relatedness matrix. So if Y is our trait, the variance in that trait is equal to the variance explained by the genomic relatedness matrix plus the environment. This is sort of your classic heritability equation. The additive variance, which is your genetic variance, divided by the phenotypic variance would give you the heritability. So we want to know, we can solve how much variance is explained by the GRM using tools like GCTA, which have been applied and validated in humans. They haven't seen much application in wildlife to date. And so we did this study, actually an undergrad in my lab did the study, and we looked at about 10 different traits in white-tailed deer, and we wanted to know whether or not they were heritable. So we had a data set of around 500 animals with genome, in this case RAD-seq data, and phenotypic measurements. They were from this island off the coast of Quebec, shown here. It's a fascinating island that was bought by a French chocolatier in the 1800s, and he tried to make it sort of his personal zoo, and he introduced a variety of species. One of them was white-tailed deer. And so a lot of research has been done on this island, and we have an amazing data set where we have both phenotypic and genetic data for thousands of animals. And so when we look at the relatedness of animals on this island, our histogram shows us a relationship like this. So this is our GRM, or the genomic relatedness matrix of both male and female and male. And the important thing I wanted to show you, and this is really the power of this approach in wildlife, is that most individuals in our data set are in fact unrelated. Here you see 1 or 2 individuals are 0.5, so that would be some mother offspring or father offspring relationship in our data. But very few of our individuals are highly related. And then this is actually desired when it comes to how GCTA works. We don't want a ton of variation or variance in our GRM. And so one of the filtering approaches you would actually do is remove highly related individuals from your data set. And so this is ideal. We can sample this pretty readily in nature, and it's in contrast to a pedigree approach, which requires tracking individuals and having a ton of related individuals. You'd actually move to the opposite end of the relatedness matrix. So we generate our GRMs, we have our phenotype, and now we can estimate heritability. And when we do that in white-tailed deer, and heritability shown here on the right, we find that most of the traits we measured, for example, hind foot length, peroneus muscle mass, above 50 to 60% heritability. So most of that variation is explained by genetics. And this is consistent with the literature. And it's consistent with humans. You know, humans, 60 to 80% heritability for things like body size and height. When we look at antlers, lower heritability, but we do see things like the number of points having, you know, above 0 heritability. The standard errors here are quite high, but this is actually quite common for wildlife. Very few studies I've seen, even with pedigrees, would have standard errors that don't allow for the estimate to overlap 0. But the important thing here is, with 380 individuals, no pedigree data, we can reliably estimate heritability coefficients that make sense. And so this is a pretty cool thing that we can now do with SNP data. So we can check off that these traits are heritable. The next question we've been interested in, now that we know they're heritable, is trying to assess the genetic architecture underlying these traits. What are the genes controlling for antlers? How many of them are there? And so what we've done to address this question is, in our database of, in this case, 20,000 deer, we sampled the extreme phenotypic ends. We made artificial pools of big antlered animals and small antlered animals. And we did this because by sampling the extreme ends of a phenotype, you should increase the additive variance. And so we're really maximizing our ability to pick up the genes underlying that trait. We don't have enough money to sequence all of the genomes. So what we did was we took a pooled approach. And this is what is known as pool-seq, where all of the big antlered individuals were pooled together and all the small antlered individuals were pooled together. We lose the individual identity. And so everything gets mixed into a single test tube. But the allele frequencies we generate, if you have high enough coverage, should be reflective of the population. And if you're not familiar with pool-seq, there are a ton of applications to this. And for example, GWAS or QTL mapping are both appropriate. And so this is a way, rather than sequencing, you know, as Cardos would have suggested in 2016, the need to sequence 300 individual genomes, now we're pooling those genomes into one pool. And there are a variety of applications, and so you can check this out. This is an old paper by Schlatter et al. But one thing I wanted to highlight is sequencing depth is important. And so we have 2 papers that are looking at this now. And the reason sequencing depth is important is if we're saying that the pooled frequencies represent that population, we need to have high enough sequencing depth to eliminate sort of that stochasticity. In other words, if you sequence to 50x, so I sequence every site 50 times, and there are 4 A's in one population and 5 A's in another population, that's a difference of 2%. Biologically, that's not relevant, but 2% some might argue is high. And so you need relatively high coverage so you have confidence in those allele frequencies, and you can eliminate that stochasticity, and you're more comfortable with your allele frequency estimates. So we had our pools, and just to show you what they look like, it's pretty neat. On average, our extreme phenotypes were all the exact same age. But when we looked at our large antlered pool, they had about 10 to 11 points on average, where our small antlered pool had 2 points. Exact same age, but massive phenotypic differences. Likewise, if we look at body length, our long-bodied individuals were, 220 centimeters on average, where our small-bodied individuals were 170 centimeters on average. So extreme phenotypes, and we're sequencing the genomes of all the individuals in these pools. And our goal is to get at the genetic architecture and identify genes underlying these traits. And when we do that, what we do is we can identify outliers or divergent regions, and we start to see patterns like this. It's a little bit noisy, but if you're familiar with outlier plots, we're starting to see what is consistent with regions of the genome that are being pulled up when you compare the traits. So here we're comparing large antler to small antlers. We see the genic position here on the x-axis, and there seems to be a region here that is highly divergent between the two phenotypes. And we like to see this plot like this because we expect there to be some linkage, and so it should be sort of a window that gets pulled up. Same thing with body size. We start to see windows or regions between large and small bodies getting pulled up. And so we generate lists and lists of divergent regions like this, and we can use this information to identify gene ontology pathways and identify sort of key molecular mechanisms that are divergent between these two regions. But some cool things we found doing this that are consistent with our understanding of antlers, for example, is we know antlers exploit oncogenic pathways. So if you're not familiar with deer, this is bone, they're antlers. They invest an incredible amount of resources in them every year, but they shed them and regrow them. And the way they do that is they exploit cancer genes, more or less. It's uncontrolled or controlled growth. And these antlers are also important for sexual selection and reproduction. And when we look at our outlier genes, what we start to see are a lot of cancer genes coming out, which is consistent with the biology. So tumor suppressor genes coming out, genes identified linked to velvet. And so early on in the season, these are actually have velvet covering them. And also we see genes that are different between antlers that are associated with sexual selection. And this MTMR2 gene, which is pretty interesting, has been linked to litter size and plays a role in spermatogenesis. And so if we're thinking about sexual selection and divergent between these two phenotypes, it makes complete sense that a gene like this would be diverging. So a cool, relatively inexpensive way to get at the genetic architecture underlying these traits. So with respect to SNP-based approaches, I've shown you that they're heritable, and there are ways to estimate the genetic architecture without sequencing hundreds of genes. Where we're going with this is I'm quite interested in this, the GRMs and heritability, and I've been simulating different scenarios to try to assess the limitation and offer guidance. How many individuals do you need? How many SNPs do you need? In our study, we had just under 400. And so I want to see, using simulations, what would be the desired sampling size and number of SNPs. So I've been working on that. All of our outliers that we've identified with antlers, we try to validate them. And so we then develop qPCR assays, and we try to validate those allele frequencies. And in fact, then we can actually quantify their effect size with respect to that trait. And we're also applying this to divergent migratory strategies in deer. And we've seen some very interesting patterns where if we restrict outliers that are only found in animals that have divergent migratory strategies, and when we compare those outliers, they're not significant in the same direction, we get a variety of candidate genes that are associated with migration in deer. And the cool thing about this approach is, and we can't quite get our head around it, but most of these outliers, believe it or not, are on exons, coding regions. So we seem to have picked up highly divergent regions between migratory strategies, and most of those mutations are on coding regions. Again, this was done using pool-seq, so we pooled populations based on their migratory direction. So that's part one. That was SNPs. I think I'm doing okay for time. I want to tell you about SNP alternatives now. And so I've already spent, the first little bit of this lecture telling you why we shouldn't look at SNPs, and then I told you a bunch of SNP stories. But I wanted to show you some work we're doing on DNA methylation because I think this is pretty cool and pretty accessible. And so DNA methylation, if you're not familiar with it, generally we focus on methylation with respect to epigenetics because it's pretty easy to quantify. And what we know is once you have areas that become methylated, that will suppress gene expression. Generally speaking, increased methylation turns off genes. And most of the cytosines that get methylated are in what are called CpG islands, areas where you have C and Gs next to each other. And so methylation, very useful mechanism our body exploits to turn genes on and off. And the way we typically quantify methylation is through our standard sequencing approaches. It can be Sanger sequencing, it can be Illumina sequencing, but we do a bisulfite conversion. And so what this means is any methylated cytosine, the methylated C doesn't change. However, if you have an unmethylated cytosine, when you do that bisulfite conversion, it gets turned into a uracil, and then when we sequence it, that comes out as a thymine. So if we compare that thymine back to the reference genome, we say, oh, there's a T, but the reference is a C, we know that T is reflective of an unmethylated cytosine. Any C that you sequence is a methylated cytosine. So this has been one of the ways in which you can estimate or quantify methylation. And so we've had two questions where we've been using methylation as a tool to answer these questions. And the first one is, does methylation and variation in methylation underlie phenotypes and population structure? And more recently, we're interested in this question about the role of methylation as animals age. Now, this first question, we've been asking questions about genetic variation in lynx for a couple years. And lynx are an interesting species to look at methylation because when we look at their genetics, at least across Canada, they're entirely panmictic. So if you ran a structure plot, they're all going to assign to the same population with the exception of Newfoundland, which is this island on the East Coast. So thousands of kilometers from Alaska to Quebec, 4,000 kilometers, genetically assigned to one population, completely panmictic. So not a lot of genetic variation when we look at SNPs, when we look at microsatellites. However, we do know that there's quite a bit of phenotypic variation. And so, for example, female lynx, their body size is different depending on the annual temperature. In addition, body size in lynx is also dependent on their prey. So here we have two environmental stressors that we know are linked to phenotypic variation, prime candidates to look at methylation. And so we've been interested in trying to get at methylation patterns in lynx. And so to do this, we sampled lynx from across Canada. These were our samples in Alaska. And we ran this assay known as epiGBS. Essentially, it's a RAD-seq assay, but with bisulfite conversion. So that's how we can assess if things are methylated. And I'm happy to chat with anybody about this protocol. It's actually pretty challenging in the lab. This bisulfite conversion is really nasty and destroys most of the DNA. And so we found that there are tissues and scenarios where we can't reliably generate this assay, but we can talk about that afterwards. So we ran this epiGBS assay, and we were able to identify methylation sites, shown here in black circles across the lynx genome. But the cool thing is we can also get SNPs from this assay. So in addition to methylation, shown in black, which gives us good coverage, tens of thousands of methylated sites across the genome of cat, we also get thousands of SNPs with this assay. And so even though our question was related to methylation, we have the SNPs, so we wanted to use them. My lab does a lot of demographic modeling, and so we were interested in that Newfoundland population. And with the SNP data here, using coalescent modeling, we could date that split, and we showed that the Newfoundland lynx here diverged around 20 to 30,000 years ago. which is entirely consistent with a potential land bridge and what we would have thought would have been the prime colonization period. So sort of an added benefit to this type of assay is you still can get usable SNP data. But looking at methylation, what we see is if we look at our SNP data, we see the same pattern where we have all of North America clustering together, and then Newfoundland being a slightly isolated population, but zero structure in North America. However, when we look at the methylation data, we start to see clearly divergent populations. So if we have methylation on unannotated regions, so just random methylation sites that we see, we can see that it clearly separates all of the populations. When we look at methylation sites on CPG islands and close to gene bodies, again, we get clear clusters of populations, and this is sort of central Canada overlapping. Huge contrast to what we see with neutral SNPs. We're able now to reliably separate populations. We can take this one step further, and we can link this methylation variation to things like climate, distance, and insularity, being on an island. And so these are the R-squared values. This is what they would call distance-based redundancy analysis, but it allows us to explain variation in methylation patterns with these environmental variables. And the cool thing is when we compare this to SNPs, there's no relationship between SNPs and these variables. And so an R-squared of 11 or even 0.09 is huge relative to the neutral genetic markers. So now we're able to actually see how variation in methylation corresponds to these variables. And again, much like the deer story, we can look at specific genes that are hyper or hypomethylated. And one of the cool things we found is when we look at the Newfoundland population, which over here we can see they actually have a small skull, so they're smaller links on average, which is consistent with the island rule. We see genes that are hypermethylated being turned on or hypomethylated being turned off. That when you look at, for example, mice knockouts, we know that that results in diminished size or modifies cell height during morphogenesis. And ZEB1, for example, larger body size in mice, hypermethylated in Newfoundland. So again, we're seeing genes that are differentially methylated that are pretty consistent with the phenotypes, and they make a lot of sense. It's a little bit of storytelling, but exciting nonetheless. So we're able to use methylation to identify genes underlying phenotypes and population structure. The next thing my lab has been doing is trying to look at methylation and how animals age. And so you may have heard of this group. They're out of UCLA. They're called the Clock Foundation, but they've developed an array that works across mammals, and it screens 40,000 methylated sites. All of these sites have been annotated. And we've been interested in using this assay to look at how methylation changes with respect to age as well biogeography. The data are pretty easy to work with. So if you've ever worked with an array data, the values you get back are between 0 and 1, 1 being 100% methylated, 0 being not methylated. We have the location of each assay has an ID. These correspond to about 200 base pair probes. We map them to our genome, and we just filter out those probes that don't map to our reference genome. And the reason we're interested in looking at methylation as animals age is because it's been shown in humans and now across mammals that there's a clear molecular clock associated with age. And so methylation increases with age, and we can use that information to reliably predict how old you are. And what's really cool about that, if you look at, for example, humans here, high correlation between methylation, but the mean error is less than 0.7. So it's accurate to within a year. And this is pretty important for humans in case of, in forensics, in postmortem incidences. But in wildlife, we have a really difficult time aging animals, and we often rely on teeth, and I'll show you some of our slides with respect to that. And so this would be actually a more accurate way to estimate age in wildlife. And so here's marsupials, again, high correlation, mean error less than half a year, probably more accurate than were you to do teeth. And so we've been interested in doing this on across a variety of mammals, and I want to show you what we've been doing with shrews. And to do this type of work, you first need to age the animals. All of these require knowing the true age to first build these models. And so this isn't easy to do with small mammals. Shrews on average live to about 18 months. So what we do is we look at tooth wear and skull morphology to age them. And generally we age them in 5 classes. And so this is what we've been doing in the lab. And one of the cool things that we do to validate our age, this is based on tooth wear, is we see that the skull shrinks, becomes shorter with age. So these are age on the y-axis, skull length on the x-axis, and you can see that the younger you are, the longer your skull is. That's what we expect for shrews. There's this real shrinkage of their skull, but also an increase of weight over time. And so we were pretty happy with our age estimates when it came to shrews. We then had 50 shrews run on the array, and we build our clock. And it turns out, lo and behold, we can reliably predict the age of shrews using only 30 sites on average. And what was really cool about this study is we actually had fetal shrews. So we had time 0 in our model, and we had adults. And so we can really help resolve some of these lower age classes. We use this approach called elastic net regression. This is really ideal for large data sets with few observations, and it's kind of the classic approach to biomarker identification. But the cool thing here is we screen 40,000 methylated sites, but we only need 30 in the end to build these clocks. And when we build these clocks, we can also look, again, the same thing we do with all these genomic studies, look at the genes that are hyper and hypomethylated. So red would be hypermethylated, blue would be hypomethylated. We can see that there's quite a few that change with age. We can also integrate everything into a gene ontology analysis. And we see, for example, that genes involved in anatomical structure become methylated with age. That would make sense. Once you stop growing, maybe you want to shut down those genes involved in anatomical structure. In contrast, we're seeing things like mammary glands increased, oh, sorry, I had that backwards, decreased in methylation. And so again, something that's consistent with aging. So really exciting stuff. Same approach we take with genomics and SNPs, but now we're doing this with methylation. And so what we can say, or what I've shown you, is that methylation, we can definitely link that to phenotypes and population structure. And we're now able to see a role of methylation as animals age. Where we're taking this, and I think this is going to be perhaps revolutionary, as I told you in shrews, for example, we just need 30 of those markers to identify the age. And so I'm trying to develop assays using Luminex technology, where we can develop 30 marker assays and take them to the wild and then start getting demographic information from bear, deer, mountain goats. So we can take their methylation and get information on age from an unknown sample. I should also tell you, we get sex as well from methylation data, and so it's quite informative there. As well, we're going to be looking for disease biomarkers, and we're interested in seeing how methylation changes with chronic wasting disease, and this might be a better assay for picking up the arrival of that disease. So moving beyond SNPs and outliers, just to sort of summarize, I've shown you what you can do with SNPs that isn't really the traditional approach, as well as some alternatives with methylation. I think for interest of time, Ella, maybe I'll stop there. I do have a bit more slides on cheap genomes, but maybe that would be better for a discussion. just don't.
As you wish. You can use another 5 minutes if you want.
Okay, maybe I'll tell everybody a little bit about some effort I've been doing to, oops, how do I get back to that? To generate cheap genomes. And this has been an interest of mine. For example, in Canada, the average federal grant we get is around 30,000 Canadian, and that is what we have to use to pay students and pay for our research. And so doing genomes can be quite expensive. And so I've been quite interested in trying to find ways to get around the cost. So these are my acknowledgements, but just to tell you a little bit about this approach. So if you do a standard library prep for whole genome sequencing, it's about $130 Canadian. And this is what Illumina sets. This is a standard cost. And so just to do libraries, this doesn't even include sequencing, you're looking at tens of thousands to even hundreds of thousands of dollars, depending on your size. And so this is cost prohibitive at scale. In addition, my lab has developed a real distaste for RADseq library preparation. I didn't mind it when I did my postdoc, but my lab does not enjoy it at all. And I recognize that. And there is this issue that it's likely only sampling less than 1% of the genome. And so I've been interested in looking at homemade alternatives to sampling the whole genome. And there are some pretty available and affordable enzyme-based approaches out there. There are some considerations with enzyme-based approaches, but the one that I've been really excited about was developed by a group in California, where they rely on what they would call ATAC-seq, but enzymatic methods that are highly diluted. And as a result, if you highly dilute these enzymes, it gets the cost of library preparation down to about $2.50 per sample and a lab time of under 3 hours. So in Canada, we have this at $4 per sample, but certainly under 3 hours. Really, really efficient. And it covers the vast majority of the genome. So what basically any chromatin accessible regions, it's going to more or less sequence. And I've been interested in doing this with some collaborators here at Trent on cattails. So I'm a mammal person, but I like cattails because they have a small genome, only about 300 million base pairs, and they're quite important for wetlands. And what we've seen in North America is our native species, latifolia, is being threatened by the non-native angustifolia. So the angustifolia is spreading, and in fact, when they hybridize, this hybrid is quite successful as well and can come to dominate wetlands. And currently, my collaborators here at Trent, they do all of their research with 4 microsatellites. That's how they identify species. And this is it's nice that you can do it with 4 microsatellites, but it's a bit of a problem when you're interested in hybridization, because as soon as you get past F1, you're not going to be able to pick up the hybrids, and maybe even F1s are contentious. And so I was quite interested in helping them out and developing a way to generate genome-wide SNPs at cost. And to do this, we ran a modified ATAC-seq approach. And what was really cool, when we did this on a preliminary run at $20 per sample, this includes sequencing, we were able to identify 500 SNPs, high quality SNPs as well, because they were present in all of the data sets that reliably differentiate the 2 parental species. And so now we've gone from 4 microsatellites, which would probably actually cost the same to run, to 500 SNPs that can reliably differentiate the species, just using an enzymatic-based approach and pretty quick bioinformatics. The cool thing about this approach too that has me a little bit excited is this ATAC-seq approach. What it does is it targets areas that are accessible. So if we think of epigenetics and things getting rolled up around histones. What it does is it targets areas that are accessible. And so you can start to compare the epigenomic landscape. You don't even need to look at SNPs. So it's your added bonus to SNPs is you can see which regions of the genome are accessible. And I've been thinking a lot about how this is probably quite important in maintaining species and hybrid dynamics. And so that's sort of the added bonus. We can get SNPs for $20 a sample, but we can also look at comparative epigenomics, which has me really excited. And you know what? We will leave it at that. And so I will go back to my acknowledgement slide and thank everybody.